I'm going to discuss about bacteriological examination of milk by BC method or breed count method. So this is one of the practical we can perform in the lab of microbiology for examination of milk. Then this practical is mostly to check the milk quality. So when it is when it is necessary to determine milk quality in a much shorter time than is possible with a standard plate count, one can make a direct microscopic count on a slide. So this is accomplished by staining a measured amount of milk that has been spread over an area 1 square centimeter on a slide. Then the slide is examined under oil and all of the organisms in the entire microscopic field are counted. So to increase accuracy, several fields are counted to get average field counts. But before the field counts can be translated into organisms per milliliter, however, it is necessary to calculate the field area. So high quality milk will have very few organisms per field, necessitating the examination of many fields. A slide made of poor quality milk, on the other hand, will reveal large numbers of bacteria per field, thus requiring the examination of few fields. So in addition to being much faster than the standard plate count, the direct microscopic count has two other distinct advantages. So first of all, the first advantage is that it will reveal the presence of bacteria that do not form colonies on an agar plate at 35 degrees Celsius. Thermophiles, psychrophiles and dead bacteria would fall in this category. Then second advantage is that the presence of excessive numbers of leukocytes and pus forming streptococci on a slide will be evidence that the animal that produced the milk has an other infection, or we also call it mastitis. So in view of all these advantages, it is apparent that the direct microscopic count has real value in milk testing. It is widely used for testing raw milk in creamery receiving stations and for diagnosing the types of contamination and growth in pasteurized milk products. So here we have the slide preparation. How to prepare the slide? It can be prepared. There are several acceptable ways of spreading the milk onto the slide, like you can see the figure given or the picture. Here we use a guide card, it is known as a guide card, to spread milk sample over 1 square centimeter on a slide, right? So the breed slide, we also call it the breed slide, has 5 1 centimeter areas that are surrounded by ground glass, obviating the need for a card. So we will proceed with the steps how to carry on with this examination. So first we will need the materials or requirements. Those include breed slide or guide card. Then we will need breed pipettes, then methylene blue, xylol, 95% alcohol, then a beaker of water, an electric hot plate, and a sample of raw milk, both poor quality and high quality. So this one, after we get everything ready, we will shake the milk sample 25 times to completely disperse the organisms and break up large clumps of bacteria. Then we will transfer 0.01 ml of milk to 1 square on the slide. The pipette may be filled by capillary action or by suction depending on the type of pipette. Then the instructor this one, we should be sure to wipe off the outside tip of the pipette with tissue before touching the slide to avoid getting more than the required amount on the slide. After that, we will allow the slide to air dry and then place it over a beaker of boiling water for around 5 minutes to steam fix it. Then we will flood the slide with xylol to remove fat globules because there might be high chance of fat globules present. So we will remove it with the help of xylol. Then after we remove the xylol from the slide by flooding the slide with 95% ethyl alcohol, gently immerse the slide into a beaker of distilled water to remove the alcohol, and we should not hold it under running water. The milk film will wash off. In case if we hold it under a running tap water, the milk film might be washed off. So after that, we will stain the smear with methylene blue for 15 seconds and dip the slide again in water to remove the excess stain. Then we will decolorize the smear to pale blue with 95% alcohol and dipped in distilled water to stop the decolorization. And we will allow the slide to completely air dry before we examine under the microscope. So we will now examine the slide under the microscope. Here, two methods of counting the bacteria can be used. That is, individual cells may be tailed or only clumps of bacteria may be counted. In both cases, the number per milliliter will be higher than a standard plate count, but a clumped count will be closer to the standard plate count. So both methods will be used. So after the microscope has been calibrated, we replace the stage micrometer with a stained slide, then examine it under oil immersion optics or objectives. Then we count the individual cells in 5 fields and record the results on the lab report. Then a field is the entire area encompassed by the oil immersion lens. So you will see leukocytes and we will record their numbers as well. Then we will count only clumps of bacteria in 5 fields, recording the numbers of leukocytes. Then we will calculate the number of the organisms, clumps and body cells per milliliter using the microscope factor. So this is the picture under the microscope examination. You will see this microscopic observation under the microscope. So that's all about bacteriological examination of milk by the breed count method. Thank you.